Let's consider protein sequence motifs. We are going to use the active site of an enzyme as an example, but keep in mind that protein motifs correspond to multiple types of functional units. If we align single-letter amino acid sequence of 3 different proteins, P1, P2, and P3, a highly conserved motif may stand out, such as the yellow boxed one here. Elvis lives. What could that mean? A subliminal message from heaven? Not likely. To get an idea, let's consider an hypothetical enzyme that uses an active site to cleave a substrate. Now fast forward into the future, and let's find 3 enzymes that descended from the first one. They will have changed in shape in most parts of the protein, except the part that was constrained by function, the active site. Now, let's align the corresponding polypeptides as represented by the colored lines. What do they share? Obviously, the sequence that they could not afford to change during evolution, the peptide forming the active site. This region is called a conserved motif. What are the implications of a conserved motif? To explain it, let's consider an enzyme whose active site we have identified because of its conservation. Then let's assume we have found mutant alleles in the population of the species carrying this gene. The mutations are missense and cause amino acid substitutions. We know the position of the mutations. We can actually predict their effect. Changes that are not in conserved regions are less likely to hurt the enzyme because they do not alter components needed for function. On the other hand, a change in a conserved region is likely to have severe consequences because it alters functional components. Conclusions: conserved proteins motifs represent protein regions that cannot change easily without losing function. We identify them through the alignment of homologous proteins. They help us predict the function of these proteins and the effect of mutations.